still chapter 2. I've already discussed how, well, briefly mentioned how important they are in terms of controlling those metabolic pathways. Okay? These hormones, insulin's one, right, they're secreted directly into blood. And they act as chemical signals, so it's very slow acting compared to the nervous system, which is very fast acting. We're talking about neurotransmitters, right? It's very fast, it's very fast. This is very slow comparatively. Okay? Obviously travel away from the cells that secreted them and specifically affect the activities of other cells or organs. Okay? Two types of hormones that we're going to talk about are steroid hormones and non-steroid hormones. This secretion is also regulated by negative feedback. Okay? And the receptors that bind these hormones can increase, that's important as well, can increase, we call it upregulation, or decrease, we call it downregulation. Locations of the major endocrine organs then. The hypothalamus in the brain does secrete hormones. And secrete what we call releasing hormones. So these releasing hormones released from the hypothalamus travel to the pituitary gland, the master gland of the body. Okay? They are actually physically connected, the hypothalamus and the pituitary gland. Okay? Thyroid and parathyroid hormones, next, thymus gland, we're not going to talk about thymus glands, we're not going to talk about the adrenal glands, okay, the top of each kidney, we are going to mention the kidney does secrete hormones, the pancreas obviously a whole battery of hormones, and we're not going to have time unfortunately to talk about the ovaries and testes and sexual hormones unfortunately, okay, or very briefly. Hormones of the pituitary gland. Right, like I say, it is connected to the hypothalamus. In terms of the anterior lobe of the pituitary gland, the hormones that are secreted there is growth hormone, thyrotropin, or otherwise known as thyroid stimulating hormone, follicle stimulating hormone, luteinizing hormone, prolactin, and adrenocorticotropin, otherwise known as ACTH. Okay? And your posterior lobe secretes antidiuretic hormone, or ADH, which we will talk about, and we won't talk about oxytocin. Okay? So we're going to talk about very briefly growth hormone. We will mention thyroid stimulating hormone, and we will mention ACTH as well. So this is hormones of the pituitary gland. In terms of the steroid hormones, these are formed from cholesterol, they're lipid soluble. They can directly affect gene activation, making a messenger RNA. Okay. The receptors for these hormones, for these steroid hormones, are located in the cytoplasm or actually the nucleus of the cell. Okay. Does affect transcription. See that? Examples, adrenal cortex. So cortisol and aldosterone are steroid hormones that release from the adrenal cortex. Estrogen and progesterone from the ovaries, testosterone obviously from the testes, estrogen and progesterone from the placenta, for example. All examples of steroid hormones all derived from cholesterol. Here's a schematic out of your book. Very simplified, so it does the job. We've got a steroid hormone that can diffuse through the phospholipid bilayer. Okay. Now, what happens then is it can bind to a receptor located in the cytoplasm of the cell. That's this blue guy right here. What this diagram doesn't show is also the steroid hormone can go through the phospholipid bilayer, through the nuclear envelope, and also bind to a receptor in the nucleus as well. It doesn't show that, but that can occur. Let's go back to this example. It's binding to a receptor in the cytoplasm. Okay? The binding of the hormone, the steroid hormone, to the receptor, that becomes what we call a transcription factor. binding of the hormone to the receptor that becomes a transcription factor. The name kind of gives it away. That complex then can diffuse through the nuclear envelope and then it can sit down on the promoter part of the gene, DNA. Okay? And then transcription occurs. And you know what the fate of transcription is, right? The negative messenger RNA. Okay. What happens to messenger RNA? Messenger RNA then leaves through the nuclear pores, embedded in the nuclear envelope, and then the rest is history, right? You know the rest. Binds to ribosomes, we get the making of protein. I'm assuming you all know about transcription and translation. You don't have to go through that, right? It should be common knowledge. Okay. Okay, so basically in a nutshell, that's the mechanism of action of the steroid hormone. Okay. In terms of non-steroid hormones, it's different. These are protein or peptide and are amino acid derived. Okay? They are non-lipid soluble. They cannot diffuse through the plasma membrane. Okay? However, a series of intracellular events, these non-steroid hormones trigger a series of intracellular events that we call second messenger systems. Example of the second messenger, we'll see that in schematic is cyclic, AMP, cyclic adenosine monophosphate. And basically, you get a whole cascade of changes that occur in the cell. Examples of non-steroid hormones are thyroxine, T3, T4 released from the thyroid gland, and epinephrine and norepinephrine released from the adrenal medulla. So, similar sort of schematic as before. Here we have a non-steroid hormone that cannot diffuse through the plasma membrane. It binds to a receptor, right? A receptor. Number B, the hormone binds to a specific receptor on the cell membrane. C, the hormone receptor complex activates adenylyl cyclase, which is bound to the receptor, and that with adenylyl cyclase combined, or it converts ATP to cyclic AMP, which is that second messenger. Okay. Fate of the second messenger, the cyclic AMP, is to activate a protein kinase. Protein kinases are enzymes. Okay. So it turns on a protein kinase, so to speak. And that protein kinase, that active protein kinase, can then make an inactive substrate active. And that active substrate can then obviously lead to cellular changes within the cell. So there's a whole cascade of events. Okay. I think that's good enough. So, those are the key points. You can read those through on your own. I do want to mention about negative feedback. A negative feedback system regulates secretion of most hormones. So what we mean by negative feedback, right? It's kind of the same idea as a thermostat. So what you've got here, I've got an example down here on the bottom right, flowchart. We've got the hypothalamus, right? And this is the thyroid hormone. It's just an example. So you've got the hypothalamus that releases a thyroid-releasing hormone. That travels to the pituitary gland, and then on that thyroid-releasing hormone activating thyroid-stimulating hormone from the pituitary gland into blood that travels to the thyroid gland, which then secretes the thyroid hormones. Now, the level of thyroid hormones in the blood is monitored by the pituitary gland and the hypothalamus. Okay? And there's a threshold. If the thyroid hormones are too high in the blood, it's a homeostatic range, right? It's too high, the pituitary gland and or the hypothalamus will shut off the release of their respective hormones. Does that make sense? And if they shut off the release of their hormones, guess what? Thyroid hormones are going to go down because the thyroid gland's not going to secrete them anymore. If the hypothalamus and pituitary deems that the thyroid hormones are low in the blood, it will continue to secrete their respective hormones, thyroid-releasing hormone, thyroid-stimulating hormone. All hormones regulated by this negative feedback system. Every single hormone is regulated and monitored by the hypothalamus and the pituitary. Also, the number of hormone receptors, like I mentioned before, can be altered, right? Upregulation increases the number of available receptors, downregulation decreases the number of receptors. If you upregulate, you find more hormones because you've got more receptors. Okay? This table here, table 2.5, this is in your book. I'm not going to go through it. I would go through it if I were you, just to get an idea on the hormone, the response to acute exercise, and the effect on exercise training. We're not going to look at all of these. Of course, we don't have time. The first hormone, or important hormone I want to talk about, is growth hormone. As you know, what does growth hormone do? It promotes muscle growth and hypertrophy, okay, by facilitating amino acid transport. It does directly stimulate fat metabolism or lipolysis. Levels are elevated during aerobic exercise. Whether it's continuous exercise or intermittent exercise, growth hormone goes up during exercise. It's stimulating fat metabolism, lipolysis, breaking down fat. Particularly important in endurance type events, right? So if I put you on the treadmill for 2.5 hours to 2 hours and assay your growth hormone, which is very easy to do, levels will be elevated because you're predominantly using fat for fuel. And to do that, you've got to break down a triglyceride molecule, and that's under the action of growth hormone. Okay. Again, there's a negative. The reason I put this slide up here, there's a negative feedback system, just like before, right? You've got the pituitary gland releasing growth hormone, and that's being monitored again by the hypothalamus and itself, the pituitary gland. Okay. In terms of the physiological effects of growth hormone, I'm going to talk about a direct effect and an indirect effect. The direct effect of growth hormone, okay, is increasing lipolysis, right here, breakdown of fat. Okay, other things. So it increases lipolysis, the breakdown of fat. It suppresses the making of fat, lipogenesis. It decreases glucose uptake into cells. Because you're trying to use fat for fuel, right? So you don't want to get glucose. You want to use glucose in the blood at a constant level. So you're trying to conserve glucose. Okay. It does increase protein synthesis in muscle, obviously, and increases lean body mass. It also increases production of IGF-1. Now, I want to talk about the indirect effect of growth hormone. Growth hormone can bind to receptors in the liver. So this is the indirect effect right here. Growth hormone receptors in the liver. It can bind growth hormone, and the liver, in response to binding growth hormone, can produce insulin like growth factor 1, IGF-1. What does IGF-1 do? It can increase protein synthesis. It can increase chondrocytes. What are chondrocytes? You've got osteocytes, which is bone, and chondrocytes, cartilage. Right? Cartilage cells. So you can increase, certainly all the growth plates fuse, right? You can increase the cartilage cells, increase long bone. That's an effect on long bone. And can also increase protein synthesis as well. And we'll talk about this again on the ergogenic age when athletes take growth hormone in conjunction with IGF-1. They're trying to increase protein synthesis in particular. The growth plates are few, so they're not going to increase long bone, right? They can get a double whammy and increase protein synthesis. Okay. So indirect and the direct effect of growth hormone. Very briefly talk about thyroid glands. Iodine, T3, and thyroxine, T4 are the thyroid hormones released from the thyroid gland. Have a lot of different consequences. T3 and T4 increase protein and enzyme synthesis, increase the size and number of mitochondria, promotes rapid cellular uptake of glucose, enhances glycolysis, glycogenesis, increases free fatty acid availability for oxidation. You saw that diagram up there on the top right as the positive negative feedback I spoke about. Again, that's, remember, hypothalamus is releasing thyroid releasing hormone, the stimulating pituitary glands release thyroid stimulating hormone, it then stimulates the thyroid glands releasing thyroid hormones. Okay? So it affects, so these T4, these thyroid hormones affect metabolism greatly. When you think about this, they can actually increase the size and number of mitochondria. Okay? There's other factors involved, but they play a big role. We're not going to have time to talk about calcitonin. Here's the thyroid gland from a cat. Right on top of the thyroid gland is the parathyroid gland that releases calcitonin. And calcitonin, what does that do? It decreases plasma calcium concentration. Okay. Let's talk about the adrenal gland secretions. The adrenal glands located on top of each kidney. There's obviously two adrenal glands. Okay. You slice open the adrenal gland, then you see an inner part, which is medulla, and that's where the catecholamines are released from. Catecholamines, in other words, epinephrine, norepinephrine. And the outer part is the cortex. The mineralocorticoids, the glucocorticoids, and even the androgens. And even a little bit of testosterone is released from the cortex, the adrenal cortex. We're going to talk about aldosterone coming up. We're going to talk about cortisol. So what about the blood concentration changes of epinephrine and norepinephrine during exercise? Obviously they both go up. Around about 50% of VO2 max you'll see a sharp rise in norepinephrine. After about 75% of VO2 max you'll see a sharp rise in epinephrine. Remember, this is part of your sympathetic nervous system, your fight or flight system. You expect those to go up, right? You expect those to go up. And obviously with duration of exercise, these will go up as well. Okay, all right. Now, what I've written down here is, understand, epinephrine is an insulin antagonist. And its actions oppose those of insulin. So what epinephrine does, it prevents the release of insulin from the pancreas. That's what we mean by it being an insulin antagonist. Okay? And remember, blood glucose homeostasis, the blood glucose concentration, is achieved by means of the balance of hormonal actions. Your body, what it's trying to do is maintain a homeostatic range with normal limits for blood glucose. If it starts to drop, it will take appropriate action. If it rises, it will take appropriate action to get it back to the homeostatic range. Okay? So, where insulin stimulates glycogen storage, as you know, you need insulin to get the glucose into a muscle cell. Right? Epinephrine and we'll see glucagon, which is also released in the pancreas, stimulate glycogen breakdown. We term that glycogenolysis. You know that. epinephrine, right here, stimulates lipolysis, to break down fat. Triglyceride breakdown, free fatty acid organization, which are used by contracting muscle and therefore glucose is spared. Alright, so you're going to think to yourself, as I did when I first saw this as a student, this doesn't really make much sense because you need insulin. If you're exercising, you need to get glucose into a muscle, right? So you've got insulin going up during exercise, okay? Going up during exercise. And let's say you've got epinephrine going up. You need insulin to facilitate glucose. You need to get glucose into blood, right? However, epinephrine is stopping that. So how are you going to get fuel? How are you going to like burn fuel in the muscle? Glucose is stopped from getting into the muscle. Does that make sense? Try and get your head around it. So if you've got low levels of insulin, you're going to have problems getting glucose into the muscle cell. You need glucose for fuel. You're doing a marathon or whatever, right? But this guy is increasing lipolysis, which is a breakdown of fat. So it's freeing up then free fatty acids and the glycerol to be used for fuel. Beta oxidation, right? Okay, I get that. That would make sense. Fine. You see that epinephrine is going up to essentially VO2 max and duration. This is 180 minutes, right? That's 3 hours. It's going up. Okay. We're predominantly relying on fat. I can see that, but it's still antagonizing insulin, blunting insulin levels. So we'll come back to that in a second. Okay. As to why it's doing that. It's doing well. I'll tell you why it's doing it. It's trying to conserve blood glucose. Remember I said that your body is trying to conserve blood glucose levels. The brain needs a certain amount of blood, right? A certain amount of glucose to live, and our body does. And basically, it's all about conserving blood glucose levels. So it's trying to spare blood glucose levels by burning fat. But we still need to get glucose into the muscle cell. We absolutely have to during exercise. We have to do that. We'll see. So here's some real data from the lab, for instance, right? So we got, this is norepinephrine being assayed from the femoral artery, and you can see, we've got epinephrine, norepinephrine here. We go to the top, this one on the right, we've got insulin. You've got subject working at 55% of VO2 max, leg exercise. Insulin starting to go down, not sharply. And then what they've done, they combine arm and exercise for about 20 minutes. Now, this person's VO2 max is being measured, and they're at 82% of VO2 max. It's pretty intense effort. But sharp blunting of insulin, sharp decrease in insulin. And then they go back to 60% of VO2 max, they go back to just leg exercise, and insulin goes up again, and then it starts to come down again. You can see quite obviously there's a big decline in insulin. That's real data. We're not just making this stuff up. It's suppressed. Okay? And as you can see, during that 20 minutes where they combine leg and arm, boom, sharp increase in epinephrine. Sharp decline in insulin. Okay? Okay, I know. There we've got about 3 more slides and we'll finish up. Okay, so another system I want to talk about is the pituitary adrenocortical system in exercise. Okay, so what we got here is we've got stress. That stress could be psychological stress for an exam, it could be physical stress, whatever, okay? That's going to affect the hypothalamus, which releases a releasing factor or hormone to the pituitary gland that then will release ACTH, adrenoccorticotropin, that then goes down to the adrenal gland, and the adrenal gland will release cortisol. We call cortisol the stress hormone. Times of stress, it's elevated. Time of starvation is elevated. Early morning, it's elevated. It's elevated because you haven't eaten for about 8 to 10 hours, your body feels stressed, cortisol levels go up. Okay, so we term the stress hormone. It can affect liver, fat, muscle, and also the immune system. Okay, very much so. Okay. So in the fasted state, cortisol stimulates several processes that collectively serve to increase and maintain normal concentration of glucose in the blood. Again, that's important. Right? So what does cortisol do? It can mobilize amino acids from what's called extrahepatic tissues. Liver is one. Sorry, not liver, muscle is one. Okay, extrahepatic tissues. So these serve as substrates for gluconeogenesis in the liver. Gluconeogenesis in the liver. Okay. So mobilization of amino acids, stimulation of gluconeogenesis, particularly in the, I've already said that, there's stimulation of gluconeogenesis, particularly in the liver, inhibition of glucose uptake in muscle and adipose, so along the lines of epinephrine, right? Inhibition of glucose uptake in muscle and adipose, that's the mechanism to conserve glucose, and stimulation of fat breakdown in adipose tissue. So its actions are very, very similar to epinephrine. Okay. So mobilization of amino acids, stimulation of gluconeogenesis, inhibition of glucose uptake, stimulation of fat breakdown. Okay. Lipolysis. Okay, before we get much more here, I've got 2 or 3 more slides. Regulation of glucose metabolism during exercise. Glucose concentration during exercise is a balance, like I already said. Glucose uptake from the exercise muscle is released by the liver. So we've got glucagon. What does glucagon do? It promotes liver glycogen breakdown and glucose formation from amino acids. So glucagon stimulates gluconeogenesis. Epinephrine promotes glycogenolysis. And I should also say that glycogen can also stimulate glycogenolysis as well. Okay? Norepinephrine promotes glycogenolysis, also promotes metabolism to break down amino acids. Okay? Glycogen, epinephrine, norepinephrine, cortisol. Here's a schematic out of your book looking at about 180 minutes of exercise. 3 hours of cycling. It's supposed to be 3 hours of cycling at 65% of VO2 max. Fairly comfortable. Okay. A whole bunch of things are assayed. Glucose in the blood was assayed during the whole time, as were all these hormones, cortisol, epinephrine, norepinephrine, glucagon. You can see that during the 3 hours of cycling, glucose was maintained. Glucose concentration was maintained. No large swing down, no large swing up. Cortisol levels rose the first 30 minutes, then declined, and then we saw big increases in epinephrine, norepinephrine, and glucagon. And that, if you recall, that's what's controlling this. That's what's maintaining the blood glucose concentration. Okay. By turning on gluconeogenesis, glycogenolysis. From all the stuff we just spoke about. Okay. Okay, last 2 slides. So back to this blunting of the insulin. Glucose uptake by muscle. If during exercise epinephrine is inhibiting the release of insulin from the pancreas, how are we getting glucose into the muscle? because plasma insulin concentrations decrease during some active exercise. Well, one way is exercise may enhance insulin's binding to receptors on the muscle fiber, reducing the need for high concentration of plasma insulin to transport glucose. What we mean by that is the insulin receptors on the muscle cell, plasma membrane, increase. Upregulation. So you need smaller amounts of insulin to bind, and then we get glucose in. Right? That's one way. So upregulation of insulin receptors, we need less insulin, that's good. Muscle contraction. The actual contraction of muscle can stimulate those GLUT4 transporter proteins to move to the plasma membrane. And that's been shown. Okay, so muscle contraction will increase GLUT4 transporter protein to move to the plasma membrane. If it does that, then you get glucose into the muscle cell. Right? And that is the way it's got in. So even though we've got this lowered secretion of insulin from pancreas to the epinephrine antagonizing it, we can still get glucose into the muscle cell. And the last slide was just simply showing you changes in plasma concentration during cycling at 65 to 70% of VO2 max. Here's your glucose, here's your insulin, here's your 180 minutes, your 3 hours of cycling again. You can see that even though insulin is going down because of epinephrine, glucose concentration is actually going up and then being maintained because of all those other hormones that I spoke about, and muscle contraction and upregulation of insulin receptors. And on that note, we'll finish and pick up from here on about fat metabolism on Wednesday. With cortisol, epinephrine, norepinephrine, and growth hormone as well. You don't have to put that up there in big print. That facilitates fat oxidation. Okay. Those are some key points then. Let's go through those because it's kind of ties into stuff that we on Monday. Plasma glucose is increased by the combined actions of glucagon, okay, epinephrine, norepinephrine, cortisol. Those are your main hormones controlling plasma glucose. We know that insulin helps enter the cell, helps the glucose enter the cell, but declines, inhibited by epinephrine in particular during prolonged exercise. And obviously with carbohydrate reserves on low, the body turns to more fat oxidation, okay? And therefore lipolysis. Okay, now I want to get into, finish off in fact, on fluid balance. Okay, fluid balance. We're going to come back to this as well as we talk about thermoregulation in a couple of weeks. I really want to talk about two hormones. There are others, one in particular, but atrial natriuretic factor or peptide. I'm not going to talk about that in this lecture. I want to talk about antidiuretic hormone, ADH, and aldosterone. Okay? So the electrolyte balance, if you like, or fluid regulation during exercise is controlled by the endocrine system. Okay? In terms of ADH, or antidiuretic hormone, this is released from the posterior pituitary, as you recall. Okay? How it's stimulated? Hemoconcentration during exercise. What do you think we mean by hemoconcentration?
It's in the blood. Your blood plasma level?
Blood plasma levels are decreasing, right? Blood plasma, so your blood becoming more concentrated. Okay. What does that do then? It results in increased plasma osmolarity, and that low plasma volume then stimulates the release of ADH from the posterior pituitary. What does ADH do? It promotes water retention in the kidneys. In an effort to bring, if you like, the blood volume back to normal levels. The schematic coming up. Back here it is. And this is straight out of your book here. So we have this guy riding his bike, obviously let's say it's hot weather. So we've got muscular number one, if you follow through here, muscular activity promotes sweating. The sweating causes loss of blood plasma, okay? Results in hemoconcentration and increased blood osmolarity. Okay. What do you think we mean by blood, or increased blood osmolarity? And in some biochemistry texts also call it osmolality, but osmolality is osmolarity. What do you think we mean by that? As a result of hemoconcentration, right?
Maybe blood flow? Blood flow?
Not blood flow.
Concentration of ions.
Exactly, good. The concentration of particularly electrolytes, sodium, potassium, chloride, etc. Okay? The percentage, this is important, right? Not the number, the percentage of those electrolytes in that blood volume has gone up. Okay? You've lost plasma. You've lost the water, so to speak. Okay? So in some respects, it feels like it looks like it's a thicker blood, right? Hemoconcentrated. That obviously we know that blood passes through the hypothalamus, so it does the pituitary gland as well. But the hypothalamus monitors that. Okay, there's actually, as I mentioned up there, there's osmoreceptors. Okay, in the hypothalamus that monitors blood osmolarity. So increased blood osmolarity stimulates osmoreceptors in the hypothalamus. From the fore, the hypothalamus stimulates the posterior pituitary gland. Posterior pituitary then releases ADH into the blood. That travels down to the kidneys, and increases the water permeability of the renal tubules and collecting ducts, leading to increased reabsorption of water. Okay? In fact, what it does, you know the collecting ducts in the kidney, right, in physiology. All these collecting ducts, okay? What's wrapping around them? Capillaries, okay? Blow this up, and so right next to a collecting duct, that's a collecting duct for renal tubule, is, in fact I should draw that closer. It's a collecting duct. That's a capillary, okay? There's these channels called aquaporins. Okay. Aquaporins. ADH comes shooting down the capillary, opens up, binds to the aquaporins, opens them up, okay, and water can enter back into the capillaries. If that happens, blood volume increases, obviously. Okay? So aquaporins. Those are those channels. Alright. So that's ADH. Aldosterone, as you know, is a mineralocorticoid hormone secreted by decreased plasma sodium, decreased blood volume, decreased particularly renal blood pressure, and increased plasma potassium concentration. Again, just like ADH, it promotes renal reabsorption for this time of sodium. It's then followed by water. Okay. And this is real data here, looking at around about 2 hours of cycling. And you can see, this is probably in the heat, you can see here that plasma volume is decreasing. Okay. And at the same time point, aldosterone is increasing, which is what you would expect. And in fact, if you put ADH onto this graph as well, you'll see a similar sort of line, okay, as you would with aldosterone. Okay, let's talk about the kidneys. So you all know that the kidneys, they certainly strongly influence the maintenance of plasma volume and blood pressure through regulation, blood pressure regulation through release of renin. Okay? So the kidneys can release a hormone called renin that's important to us. Okay? And renin initiates what we call the renin-angiotensin-aldosterone system or mechanism. Okay? Alright, so similar to before, now we have a lady roller skating, clear free trail, right? It's hot. Okay? So again, the muscular activity promotes sweating. If you sweat, you're losing plasma volume. What does that do? Increases blood pressure. Okay? Sweating reduces plasma volume and blood flow to the kidneys. Number 3, reduced renal blood flow then stimulates renin release from the kidneys. Okay? Renin leads to the formation of angiotensin II, a little polypeptide in the blood, which is converted to angiotensin 1, which is converted to angiotensin 2. That all occurs, believe it or not, in the lungs under the action of an enzyme called angiotensin converting enzyme, or ACE. Okay. Angiotensin 1 is converted to angiotensin 2 in the lungs under the action of angiotensin converting enzyme. Angiotensin 2 can then travel in the blood, okay, to the adrenal cortex and release aldosterone from the adrenal cortex. I'm not sure your book talks about it. So angiotensin 2, okay, is also a very, very potent vasoconstrictor. So as you can see up here, with this sweating, it reduces plasma volume and blood flow, right, and therefore blood pressure. Angiotensin 2 is a very potent vasoconstrictor. The potent vasoconstrictor then is working to increase blood pressure, right? You vasoconstrict, you increase blood pressure. So that's one of its actions that actually is not on this schematic, okay? Another action of angiotensin 2 is then to travel down to the kidneys, just like ADH, and increase sodium and water reabsorption from the renal tubules. Now it doesn't open aquaporins, it binds to sodium channels, right, and lets water back in like this from collecting ducts and renal tubules. And remember, wherever sodium goes, water goes with it. It's an osmotic form, okay? So it opens up sodium channels, sodium comes back into the capillaries, and water follows. And therefore you increase your plasma volume, obviously. Makes sense? Okay? So lastly, number 6 then, plasma volume increases urine production, decreases simultaneous exercise. Okay. So those are the key points. I'll let you read those.